This is the second presentation regarding fungi, and we're picking up on the characteristics of the different four types of fungi that we're going to be dealing with.
So let's talk about this first division, the Zygomycota, or Zygomycetes, and they produce zygospores, which are thick-walled, environmentally resistant spores, and they develop from the fusion of two sexually opposite cells. Now, examples of this include Rhizopus stolonifer, which is common black bread mold. These are also used in the fermentation of rice into sake by another of the Rhizopus species, and cortisone production by yet a different Rhizopus species. So as you can see, the Rhizopus are a great example of Zygomycota, and they have a wide range of uses and impacts. We also have our Azomycota. There's something like 30,000 species of the Ascomycetes, sac fungi, the sac being the asci. Now, most of these are filamentous, that is, they form long strands, but there are some that fall into the category of yeasts, which are single-celled organisms. Those that are in long strands have septa between the cells. That is a kind of an incomplete wall, and that incomplete wall does allow the sharing of materials from one cytoplasm to another. The sexual structure is an ascocarp, and it contains ascospores. And as you can see here with this image of Canidia, you can see the ascospores. In this particular case, Aspergillus quadrillanatus. The Ascomycetes, the Ascomycota, include various penicillin species. And the penicillin have been very beneficial for us in forming antibiotics, including any of the penicillinases, anything that is a psyllin, basically. So penicillin, ampicillin, amoxicillin, methicillin, these are forms of penicillin. Some have been chemically modified, but they are derived from these fungi. A thing to be aware of, that fungal organisms produce materials that are antibiotic, that is, they kill bacteria, whereas bacterial organisms produce things that are antifungal or kill fungi. And that's just the way that they compete for food materials. But penicillin species, aside from making antibiotics, it is one of the penicillins that's involved in the production of blue cheese. There are also Aspergillus species, and these are common in household dust. They often cause allergic reactions and respiratory illness. They make some toxins called aflatoxins, which can cause some respiratory distress and other problems, but have some beneficial properties as well. They are involved in the production of certain food products, including citric acid, soy sauce, vinegar, and Beano, which helps break down galactose, which is a certain sugar that exists in foods that can cause intestinal gases. The image that you see to the right, this is penicillin. Also, among the Ascomycota, we have Claviceps purpura. This is a particular organism that has certain pathogenic capabilities. It infects rye specifically. This grows in grain foods, and what will happen is that the toxins that it produces, if you ingest them, will result in ergot disease in humans, or ergot poisoning. Initially, the symptoms may not be too bad, but they can get quite severe. We have found, though, that this is a potential headache cure in low doses, but high doses, potential bioterrorism weapon. Paranoid and angry enough that it may have contributed to the French Revolution. There was a particularly wet year just prior to that, and a lot of the grain ended up contaminated with this particular organism, Claviceps purpura, which may have resulted in a large portion of the rural population of France, at least, getting this poisoning, which, again, they turned and looked at all the wealth that their neighbors had, began burning down houses. The next division of fungi is the Basidiomycota, or Basidiomycota. These produce spores, which form in gills at the base of a mushroom, basically. So this includes the mushrooms. They are called the Basidiomycetes, as they make a basidiocarp, which is this fruiting body. In fact, what we know of as a mushroom is the fruiting body of a much larger organism. The bulk of the organism is underground. The basidiocarp, that is the portion that comes to the surface simply to reproduce. In the Basidiomycota, there is something called a dikaryotic stage. Eukaryote, it has a nucleus, and in a dikaryotic stage, there's actually two nuclei, and it's a rather long stage where we have two haploid nuclei. So it's not like our cells that have diploid nuclei, but at some point, these haploid nuclei merge to become a diploid eukaryotic cell. Again, they undergo meiosis instead of mitosis to form haploid basidiospores, and then those spores get released into the environment. Again, we have a mixture of the genetic material from one organism and the other, and you end up with basidiospores that are genetically unique from the parent, and those spores get spread, and you get new fungi. Now, the figure 8-7, and it's in your textbook, that figure shows the basic reproductive cycle of the Basidiomycota, and it's on page 160 of your text. But you can see on the underside of mushroom cap, there are gills that are lined with these club-shaped sexual reproductive structures, again called basidia. And there is fusion that occurs within the basidium to give rise to the zygote, which is a diploid cell. The zygote undergoes chromosome halving process called meiosis, and then you have haploid basidiophores, which contain genetic material from both parts. Ultimately, the basidiospores are released, and they germinate to form in sexually opposite hyphae, and you end up with a new mycelium. The mycelium grows, and later on, you have this whole cycle begin again. Once it gets large enough, it produces another mushroom-like structure. Continuing with the Basidiomycota, there are about 25,000 named species, and this includes a lot of stuff. Puffballs. I remember as a child going around stomping puffballs. There's shelf fungi, there's earth stars, there's stinkhorns, jelly fungi, gill fungi, which include some poisonous ones, such as the toadstools and other things. There are organisms that make rust disease and smut disease, and then there are edible mushrooms, which include the shiitake. The last of the divisions of fungi are the Deuteromycota, and there are about 25,000 species. Right now, there's no currently identified form of sexual reproduction. These are sometimes referred to as the Deuteromycetes or Fungi imperfecti. They do form conidia, but it is a haploid conidia. A lot of human pathogens, including dermatophytes like athlete's foot. There's also a group which is important for us, that is the Pseudomaceri, which makes chemicals that mimic the effects of insulin. And so there are certain uses there. We have some beneficial and harmful fungi, and the beneficial ones include the yeasts. We use the yeast for a lot of things, and the Saccharomyces, or as Azomycota, these are single cells, and they will ferment pretty much any sugar there is. And we can use that in a couple of ways. For instance, Saccharomyces cerevisiae is used for baking. And what will happen is, under the right circumstances, you get an accumulation of carbon dioxide during this Krebs cycle, which will make the dough rise. They're breaking down sugars, and as a byproduct, you get this carbon dioxide. They also have enzymes which will break down the gluten, which is in the starchy material that's in flours, and give a nice spongy texture to bread. There's also Saccharomyces ellipsoides, which is used for brewing. And in this case, in the absence of oxygen, you get fermentation that occurs. And the end result is the byproduct of carbon dioxide and ethanol. Depending on which starting materials you use, you end up with wines, beers, whiskeys, whatever. Now, yeast have other activities beyond bread making and fermentation of alcohol. They are used as tools for biotechnology. In fact, these days the vaccine for hepatitis B is produced by modified yeast. We've modified their DNA in such a way that they are producing the necessary protein that will activate our immune systems. This has also been the yeast have also been a tool for the Human Genome Project and have aided in discovering the sequencing of the components of DNA. A last group, which is kind of interesting, are the lichens, because they are an interaction between a fungus and a photosynthetic partner. So what we end up with is this symbiotic relationship. Almost all of the lichens have Astromycete as the fungi. Most frequently, we have photosynthetic partners, which include Trabucci, which is a green algae, Trentophilia, which is a different green algae, and we have Gnostic, which is a cyanobacterium. The thing is that we get a mutual benefit because the fungi protects the algal structures. The algae are able to provide nutrients for the fungi. So the end result is that lichens grow in a diversity of environments. They can grow in the desert and in the Arctic zones, on bare soil, on tree trunks, rocky areas, and the photosynthetic partner provides sugar and carbohydrates for the use of the fungal partner. The fungal partner supplies protection and some basic biochemical building blocks in that they are breaking down some rocky formations and chemical formations. They don't require a lot of water, which is surprising given the percentage that most organisms are of water. So they can survive on as little as 2% water. End result is that there is some very slow growth. They grow in areas where very few other organisms can grow, so there's not that much competition, but their growth is slow, generally less than 10 millimeters per year. And because of this, we can actually use the size of a lichen to estimate the overall age of some artifacts.
We're going to stop at this point and pick up with a continued discussion on fungi on our next part of the presentation. Thank you.